So first of all, I would really like to thank the ISHR for initiating this webinar. I think this is fantastic, especially under the current circumstances. And usually I would work in the James Black Center here at King's College London, which you see on this first slide on the second floor. But of course, like many of you, we're all currently working from home. So what I would like to do in this half an hour, I would like to give you some insight how we use multi-omics technologies to study cardiovascular disease. And as you all know, cardiovascular disease is a prime example for a multifactorial disease. So it's not just about genetic predisposition. The disease etiology is, of course, equally determined by environmental and by lifestyle factors. So we need to extend our knowledge beyond the genome and include additional layers of molecular information, such as the transcriptome, the proteome, the metabolome, and the lipidome, to get a better understanding of the interactions between genes and environments. Now, while we have a wealth of genetics data, we still have relatively few data on corresponding protein and metabolite levels. And we really often use genetic information as a proxy for protein function. But ultimately, the proteins are the molecules of life, and this is the molecular machinery that mediates disease. So in addition to studying genetic variation that gives us the blueprint for the proteins, we also want to study the proteins directly. And I think all of you are aware that the human genome encodes for approximately 20,000 protein coding genes, but it is estimated that due to alternative splicing, post-translational modifications, the human body may well express around 100,000 to 500,000 different proteins. Now, to give you an overview of the presentation, I've structured it into three parts. First, I would like to give you some background on cardiovascular risk assessment and what has changed since the time I left medical school 20 years ago. Then I would like to summarize some attempts for omics profiling, including our own data, but also data from other groups in clinical cohorts. And finally, I would like to share with you some very novel insights relating to PCSK9. And I'm sure most of you will be aware that PCSK9 is one of the latest, most promising treatment targets to lower LDL cholesterol. Now, cardiovascular risk assessment is still pretty much relying on very simple parameters. So these are personal details like age and gender, your clinical characteristics such as family history, smoking, diabetes status, blood pressure, but relatively few molecular measurements. In fact, for lipid measurements, we rely on low-density lipoprotein cholesterol, the LDL cholesterol, which is the bad cholesterol, and high-density lipoprotein cholesterol, which is supposedly the good cholesterol, and total triglyceride levels. So these measurements haven't really changed over the past few decades. What has changed, however, is that we are consistently aiming for lower and lower LDL cholesterol levels in order to prevent cardiovascular disease. And the other major paradigm shift was that HDL cholesterol, although it's a very good biomarker and it's inversely associated with cardiovascular disease, it turned out to be a rather more difficult treatment target. And it is now thought that HDL cholesterol may not be causal for cardiovascular disease. And this has led to a shift in the intention from HDL cholesterol to triglycerides. Triglycerides, total triglycerides, do not inform treatment decisions unless they are usually very high, but HDL cholesterol levels are known to be inversely associated with triglyceride levels. And while HDL cholesterol may have been the better biomarker, it could have been that triglycerides would have been the better treatment target. Now, some of this is supported again by genetic studies, and I would like to point out these two studies, one from Denmark and the other one from the United States, where both of these studies looked at a loss of function mutation that is associated with lower triglyceride levels. So this is a loss of function mutation in an apolipoprotein called Apolipoprotein C3. It's a relatively rare mutation, but they had a large cohort, and in heterozygotes, they found that this loss of function mutation is associated with a 44% reduction in triglyceride levels. Importantly, this reduction in triglyceride levels also was associated with a profound reduction for the risk of coronary disease. And here you can see the corresponding data from the NHLBI cohort, where we see a 40% reduction in the odds ratio in people carrying this loss of function mutation in apolipoprotein C3. So again, this provides some genetic evidence that triglycerides may actually also play a role in cardiovascular disease. And in our clinical studies, however, because of the major focus on LDL cholesterol and HDL cholesterol, most studies to date only measure two apolipoproteins. And this is, of course, ApoB 100. This is the apolipoprotein of LDL, and ApoA1, which is the major protein on HDL particles. It constitutes up to 70% of the entire protein content of HDL. So just a brief snapshot. I think many of you may be aware that, of course, lipoproteins are consistent out of a phospholipid monolayer. This phospholipid monolayer shields the triglycerides and cholesterols, the hydrophobic lipids within these lipoprotein particles. And the structural apolipoproteins, these are the ones that basically mediate the function and to determine the uptake and the turnover of these lipoproteins. Now, there are, of course, many other apolipoproteins that are bound on lipoprotein particles. However, they are usually not measured. So in this first part of my presentation, I want to show you how we use mass spectrometry-based profiling in clinical cohorts to extend the measurements of apolipoproteins beyond ApoA1 and ApoB. And the way we do this is not by using antibody-based assays, but by using mass spectrometry. And I think one reason is that whenever we do protein research, we rely on the quality of our binders. And usually we have antibodies that we trust, and some of these antibodies are very reliable. But as we also know from our own experience, many antibodies, of course, can vary in their quality. First of all, for not all human proteins that are encoding in the human genome, you can get high-quality antibody. And furthermore, antibodies do not recognize a protein. They only recognize an epitope. They only recognize a part of a protein. And I'm sure you have come across circumstances where you used different proteins, different antibodies against the same proteins, and surprisingly often get different results. So with mass spectrometry, on the other hand, we do not rely on binders. We measure proteins directly. And to be accurate, we don't measure proteins. We use multiple peptides from the same protein as a readout for its quantitative abundance. Now, this is a very simple scheme of the workflow for a mass spectrometry experiment. I just want to point out that we really use minute amounts of plasma. So 10 microliter of plasma is sufficient to measure hundreds of plasma proteins by mass spectrometry. And when we do clinical studies, we always add heavy reference peptides, and this allows for more accurate quantification, because we compare the heavy peak from the reference peptide against the light peak from the endogenous peptide in the plasma sample. And these protein assays are run simultaneously, so you measure hundreds of proteins in any one run in one sample. Now, this type of technologies we then apply to clinical cohort, and in particular to the Bruneck study. So I graduated from the University of Innsbruck, and the University of Innsbruck was organizing this study in the 1990s. This is a longitudinal study with now 30 years of follow-up. We have serial sampling every 5 years. And this is very important because we can determine protein levels before the onset of disease, and we have less confounding by comorbidities and medication as you usually have when you just use case control studies. And Bruneck is shown here on the right. It's a small town in northern Italy. And one advantage of this study is that because the scenery is so beautiful, very few people actually leave. So this study has exceptional follow-up rates of up to 98%. Now, here are the results for the associations of apolipoproteins with incident cardiovascular disease. So on the y-axis, you see the different apolipoproteins that were measured at this particular time. And on the x-axis, you get the hazard ratio for a 1 standard deviation higher concentration of each of these apolipoproteins. And what you can see immediately is, of course, what would be expected. ApoB 100 is predictive of incident cardiovascular events such as heart attacks and stroke. You get a hazard ratio of 1.3, so there's a 30% higher risk associated with a 1 standard deviation higher concentration of ApoB 100. However, if you look at the other apolipoproteins, you see that ApoE, ApoC3, and ApoC2 actually show a similar strength in association with cardiovascular events than ApoB 100. And in fact, in some of these cases, this association was even stronger. And what that suggests is that there is cardiovascular risk probably beyond LDL cholesterol. LDL, the lipoprotein that carries cholesterol, predominantly has one apolipoprotein, ApoB 100, whereas other apolipoproteins, such as the triglyceride-rich lipoproteins, such as VLDL, they also carry ApoB 100, but in addition, they carry additional apolipoproteins. And in our analysis, ApoC2, ApoC3, and ApoE very nicely correlated with triglycerides, suggesting that in plasma and total plasma, their predominant association is with the triglyceride-rich lipoproteins. Now, what do these apolipoproteins do? So this is a simple scheme again of a very low-density lipoprotein. It has ApoB 100 as an integral membrane protein, but then it has associated apolipoproteins, and that includes apolipoprotein E, ApoC2, and ApoC3. Apolipoprotein E is required for the uptake in the liver. Apolipoprotein C2 and C3 are activators and inhibitors of an enzyme called lipoprotein lipase. And lipoprotein lipase sits on the surface of endothelial cells. And when it gets in contact with the triglyceride-rich lipoproteins, it hydrolyzes the triglycerides. And this becomes very visible if you actually look at plasma samples before and after a meal. So in a postprandial sample, you have got this turbid solution. This is because of all these triglyceride-rich lipoproteins that are circulating. But these are cleared very rapidly by enzymes such as the lipoprotein lipase to basically restore the plasma sample to a more translucent fluid. And as I mentioned before, loss of function mutations of ApoC3 have been associated with lower triglyceride levels. Again, from this schematic, this becomes obvious. ApoC3 is an inhibitor of lipoprotein lipase. So if you have the loss of function mutation of an inhibitor, you get more activity of the enzyme and therefore lower triglyceride levels. And this, as I pointed out previously, was associated with a reduced incidence of cardiovascular events. So what I wanted to show you here is how we have used, on one hand, a proteomics-based approach to demonstrate that triglyceride-rich lipoproteins may be underappreciated in mediating cardiovascular risk. This complements a previous study where we have used in the same Bruneck cohort MR spectrometry-based lipidomics approach, and again made this surprising observation that certain triglyceride species showed a very strong association with future cardiovascular events. So to paraphrase Stephen Hawking, the cost of poor data is delusion of knowledge. I think we have to be aware of this misconception that if you measure ApoB 100, you're not only measuring LDL, you're measuring all the ApoB 100 containing lipoproteins. And this is VLDL, IDL, and LDL. Chylomicrons carry a truncated form of ApoB, and this is ApoB 48. On the other hand, when we measure ApoA1, we are not just measuring HDL, but we are actually not discriminating between the different size of the particles of HDL. And we now know that many of these have very different biological function. So unlike in a proteomic and a lipidomic approach, where we look at the protein content or the lipid content, there's another approach called nuclear magnetic resonance spectroscopy, where you can look at the size of the different lipoprotein particles. And here I would like to share with you some data that were published by a group in Oxford. The first author was Michael Holmes, and they used nuclear magnetic resonance spectroscopy in the China Kadoorie Biobank. Again, what this technology allows you to do is to measure the different lipoprotein particles and their sizes. And this is the summary of their finding. And it nicely complements this data I've just shown you from the Bruneck study. Again, cardiovascular risk, and in this case, they looked at the incidence of myocardial infarction and ischemic stroke, was not only limited to LDL, it extended to all the ApoB 100 containing lipoproteins such as VLDL and IDL. On the other hand, HDL cholesterol, as expected, was inversely associated with the risk of myocardial infarction and stroke. But this inverse association was only seen for large and medium-sized HDL particles, but not for small HDL particles. And again, the other surprising observation, if you look at the triglyceride content in green, regardless of where the triglycerides are compartmentalized and in which lipoproteins they are present, they were always positively associated with the risk of myocardial infarction and ischemic stroke, whereas there was a null association, as one would expect, with the incidence of intracerebral hemorrhage. So what this really alludes to is that there may be additional cholesterol-containing particles that contribute to atherosclerosis. We know that low-density lipoproteins are the main carrier of cholesterol in the circulation. This is indicated here by this pie chart. They're cholesterol-rich and triglyceride-poor. They're also small in size, and they can cross from the plasma into the arterial wall and therefore contribute to atherosclerosis. On the other hand, the triglyceride-rich lipoproteins, such as chylomicrons or VLDL, they are much larger, they're triglyceride-rich, and they are thought that they can't cross easily from the plasma into the arterial wall. However, when these triglyceride-rich lipoproteins are circulating, they're exposed to this enzyme lipoprotein lipase. And like a balloon that is deflated, the hydrolysis of these triglycerides within the lipoprotein particles makes these triglyceride-rich lipoproteins smaller in size, and they're then called remnant particles. Now, these remnant particles still carry some triglycerides, but they also carry now a lot of cholesterol, and they're in size similar to LDL, and it is thought that not only LDL can cross into the arterial wall, but also these remnant particles of triglyceride-rich lipoproteins. So the question is now to what extent the cholesterol buildup in arteries is only due to the effect of LDL depositing cholesterol in the vessel wall, or also due to the remnant cholesterol that originates from triglyceride-rich lipoproteins. So to conclude the first parts, I still find it surprising that clinically we do not have many measurements to assess cardiovascular risk apart from total cholesterol, total triglycerides, as well as HDL and LDL cholesterol. Now, cardiovascular risk is clearly determined by LDL cholesterol. It's the major risk factor, but the risk may extend to other ApoB containing lipoprotein particles, such as VLDL. And this may become more important in an era like the statin era, where LDL cholesterol is very well managed. So to some extent, the residual cardiovascular risk we see in these patients may come from the triglyceride-rich lipoproteins. It's a completely different story for HDL cholesterol. HDL cholesterol is a very promising biomarker, but now evidence from genetic studies, as well as the negative results from clinical trials, would suggest that low HDL cholesterol is not causal for cardiovascular disease. And this has generated a renewed interest, on one hand, in triglycerides, on the other hand, in HDL functionality. So there's some very nice example from Dan Rader's group, where they now look at cholesterol efflux capacity rather than HDL cholesterol. And as I've shown you with the NMR measurements, investigators are looking at the different HDL particle sizes. So this brings me to the second part of my talk, and this is where I would like to share with you some data where we obtained novel insights relating to PCSK9. Now, PCSK9, I'm sure many of you are familiar with this particular protein, but just to recap, PCSK9 is a secreted protein, and it regulates the numbers of LDL receptors on the hepatic surface. PCSK9 binds to the LDL receptor and facilitates its uptake and targets it for lysosomal degradation. So therefore, the current therapeutic strategy is to inhibit PCSK9. Currently, this is done by monoclonal antibodies, but there's also new clinical trials on the way where they use antisense technology. And the idea is that if you remove PCSK9, you get more LDL receptor surface expression and therefore more clearance of LDL particles from the circulation. And this has been proven extremely successful, and you can now reach target levels of LDL cholesterol that were not achievable by statin treatment alone. Now, one interesting fact about circulating PCSK9 is that not all of the PCSK9 is circulating freely. Actually, the majority appears to be protein bound. And there are two main candidates for PCSK9 binding. The first one is LDL, and that makes a lot of sense because PCSK9 regulates LDL levels. And this is a very nice paper in the Journal of Biological Chemistry, where they demonstrate that binding of PCSK9 to low-density lipoproteins inhibits PCSK9-mediated LDL receptor degradation. And the other candidate is a peculiar LDL particle, which is called lipoprotein a. So it's an LDL particle that contains an additional apolipoprotein called here apolipoprotein a. And again, this is an emerging risk factor for cardiovascular disease. It's thought that 1 out of 5 people have very high levels of Lp(a), and it seems that this particular LDL particle is very atherogenic. It's not quite understood what mediates these atherogenic effects, but again, there are ongoing clinical phase 3 studies as outcome trials to show whether Lp(a) is not just a biomarker, but actually a novel treatment target. Now, in the Bruneck cohort, we also performed nuclear magnetic resonance spectroscopy, again, using these magnets to separate lipoprotein particles by their size. But we then did one additional correlation where we looked at the association of these lipoprotein particles with plasma PCSK9 levels. And this revealed some very surprising associations. On one hand, we saw that PCSK9 plasma levels were correlated to pro-atherogenic lipoproteins, as one would expect. But we also found a positive correlation with anti-atherogenic lipoproteins such as HDL. And this is shown here. It's a bit of a complicated slide, but just to show you this data, on the x-axis, you've got the different lipoprotein subfractions. So each lipoprotein has different sizes, and that's abbreviated by XXL, XL, L, medium, and small. And on the other hand, you've got the lipid content of these lipoprotein particles. And we show here the Spearman correlation. So red indicates a positive correlation, blue a negative correlation. And we get the strong positive correlation between plasma PCSK9 levels and the circulating levels of ApoB containing lipoproteins, which you would expect if PCSK9 regulates the LDL receptor. But we also saw this unexpected positive association of PCSK9 plasma levels with small and medium HDL. And this for us was a surprising observation, which we really wanted to follow up further. Now, to do this, we first used a sandwich ELISA where we coated anti-PCSK9 antibodies to capture the PCSK9 from the circulation, but then used 3 different detection antibodies to, on one hand, detect PCSK9 that's bound to Lp(a), PCSK9 that's bound to ApoB containing lipoproteins such as LDL, and on the other hand, PCSK9 that's bound to HDL by using an anti-ApoA1 antibody. And here are the results. So on the y-axis, you've got the intensity for the signal for PCSK9. If we use ApoA as a detection antibody, we see that only a small proportion of the circulating PCSK9 is bound to Lp(a). We get the better signal if we use an ApoB antibody, suggesting that there is a substantial proportion of PCSK9 that is binding to ApoB containing lipoproteins such as LDL. But to our big surprise, the strongest signal was actually observed for ApoA1, and this suggests that not the ApoB containing lipoprotein, but in fact HDL may be the main carrier of PCSK9 in the circulation. There's, however, one big limitation to this assay, and again, this is we are using an antibody to capture lipoprotein particles. The lipoprotein particles have different sizes, so therefore the capture may vary in efficiency. So we really thought we need an additional readout to confirm this finding, and we used a depletion method. On one hand, a depletion for ApoB containing lipoproteins, and on the other hand, a depletion method for HDL. So we generated LDL and VLDL depleted plasma as well as HDL depleted plasma, and we showed how this depletion affects PCSK9 plasma levels. So here are the results, first after the ApoB immunodepletion. So again, to remind you again, if we deplete for ApoB, we are not only removing LDL, we are also removing Lp(a) and the triglyceride-rich lipoproteins that carry ApoB. Now this depletion was very successful. So in blue, you see the non-depleted plasma. In red, you see the ApoB depleted plasma. We used, again, targeted mass spectrometry to confirm that upon ApoB depletion, we barely get any detectability for ApoB, as well as for Apolipoprotein. But in contrast, the HDL-associated apolipoproteins barely changed upon ApoB depletion. And despite this successful depletion of ApoB, plasma concentrations of PCSK9 only dropped by 20%. Now in the next experiment, we did the opposite. This time we went for HDL immunodepletion, so we depleted all the HDL particles. And in this case, again, we confirmed by mass spectrometry that we got very successful depletion of HDL, as indicated by this loss of signal for ApoA1 and ApoA2. We get some removal of ApoB containing lipoproteins, but again, this is only in the order of 50%. So although it's not entirely specific, we have very successful depletion of HDL and some removal of ApoB containing lipoproteins. But yet, if we look at the plasma concentration of PCSK9, almost 90% of the PCSK9 plasma concentration is removed after HDL depletion. So what this suggests is, as I mentioned earlier, that indeed HDL is the main carrier of PCSK9 in the circulation, and that this may be a novel function of HDL. And this is a simple Comassi stained gel, and where you can nicely see how successful HDL was depleted. So ApoA1 and ApoA2 are only present in the HDL fraction, but are completely absent in the HDL depleted fraction. And if you probe for PCSK9, you can see that, again, the PCSK9 content is almost exclusively present in the ApoA1 containing fraction. Now, previous studies that looked at the distribution of PCSK9 in lipoproteins mainly used density centrifugation to separate lipoproteins. So the triglyceride-rich lipoproteins are light, they float on the surface. On the other hand, protein-rich lipoproteins such as HDL are heavy, so they sink to the bottom. And usually the layer in between, this is where you find the majority of your low-density lipoproteins. However, it is important to point out that this density gradient centrifugation is an enrichment method. It doesn't generate pure samples. And if you look for the content of ApoA1 as a surrogate marker for the presence of HDL, then of course you find ApoA1 in the heavy fraction, but you also find ApoA1 in the medium fraction, suggesting that this LDL containing fraction is contaminated by HDL particles. So to verify our findings, therefore, we now use the ApoB depleted fraction, the heavy fraction after density centrifugation. We again performed the HDL immunoisolation, but now just not from total plasma, but only this heavy fraction. And we can nicely recapitulate our finding that again, PCSK9, as well as the furin cleaved form of PCSK9, is present in the ApoA1 containing fraction, and very little of it is found in the fraction that is depleted of HDL particles. So this brings me to the second part of the conclusion. I think these are some thought-provoking findings, because we have now provided 5 lines of evidence to demonstrate that PCSK9 is predominantly found on HDL. We used immunocapture of PCSK9-associated lipoproteins. We demonstrate that if we deplete ApoB, we only get a 20% reduction in plasma PCSK9. On the other hand, if we deplete HDL, the majority of the PCSK9 is removed, suggesting that some PCSK9 is present, for example, on LDL, but the majority seems to be carried on HDL. And we then repeated these findings with immune affinity enrichment from total plasma, as well as from plasma that was subject to density centrifugation. So what these studies that have initially started with an omics approach have revealed is that, contrary to current assumptions, PCSK9 may not be carried on LDL and Lp(a) alone, but actually the major carrier of PCSK9 in the circulation may be HDL. And of course, this raises now many questions, and I'm looking forward to some of the questions in the Q&A session. What is, of course, the functional consequence of PCSK9 binding to HDL? What are the protein interactions that are responsible for this PCSK9 binding? And most importantly, are the PCSK9 binding properties contributing to the potential cardioprotective effects of HDL, or in brief, could HDL be an endogenous inhibitor of PCSK9? So there's a lot of questions that have emerged from this study, but this is how far we have come so far, and I really would like to acknowledge all the people in my group who contributed to this work, in particular a PhD student, Jean Bernop, who is currently writing up his thesis and has just published this paper in JACC, showing that PCSK9 is the main carrier, or HDL is the main carrier of PCSK9 in the circulation. And of course, this study is not possible without funding, and in particular, I would like to acknowledge the British Heart Foundation and the Foundation Leduc, as well as our VASCAGE consortium and the contribution from all our collaborators. And with this, I'd like to end, and I'm very happy to take questions. Thanks for your attention. Okay.
I will just unmute myself now, and probably if you could, perfect. That's ideal. So I will just try and put us up on the screen together, and then that should be, if you probably just could stop sharing the screen, Manuel, as well, maybe.
Yep.
There is a red button, I think, on the top.
Okay, yep.
Perfect. And I think now the attendees, everybody in the audience should be able to see two of us side by side. I mean, first of all, thank you for a wonderful talk. Really exciting stuff. And thank you to the audience as well for participating. I mean, again, we've got fantastic numbers. So clearly there's interest for this kind of medium of presenting science, at least at this time. So Manuel, I mean, there are some questions in the Q&A box. I think there's, so I don't know if you'd like to start addressing, there's one at least for sure.
Yeah, there's one from Fabio De Lisa.
Yes.
Greetings to Italy. I think, again, this is a very important question, and it relates to chylomicron remnants. And actually, most clinical studies to date are using fasted plasma. So therefore, we expect that most of the cholesterol in fasted plasma is, of course, in LDL, whereas we have a relative paucity of triglyceride-rich lipoproteins, in particular chylomicrons. So chylomicrons are produced in the postprandial phase. They are cleared over a couple of hours. So if you use individuals that are fasted, you wouldn't find many chylomicron remnants. So the hypothesis is that remnants from triglyceride-rich lipoproteins, and these are chylomicrons as well as VLDL, may contribute cholesterol to atherosclerotic lesion development. Now, given the fact that most of us now constantly live in a postprandial state, and our periods of fasting are increasingly short, it is actually a very valid hypothesis to what extent these chylomicrons and chylomicron remnants could also be important for depositing cholesterol. And therefore, it is, I think, an interesting question, not just to look at static concentrations of triglycerides, but actually at clearance rates of triglycerides. So if you actually can't remove these triglycerides fast enough, then maybe there is enough time to build these remnants that ultimately then may deposit cholesterol in the arteries. So I think there's clearly a shift where, in addition to looking at LDL cholesterol, which is undoubtedly the main risk factor for cardiovascular disease, the question is now where's the residual lipid risk? So if your LDL cholesterol is well taken care of, is there additional risk in your triglyceride-rich lipoproteins, or is there additional risk in lipoprotein little a? And I think in the coming years, we are going to get a better understanding of how cardiovascular risk is determined, because now we have new treatment options to actually target these different lipoprotein particles.
Thank you. It seems to me that people cannot actually get the Q&A box. I'm not sure why that is the case. Everything seems to be enabled for it. So maybe if I could just ask if there are any other questions, that maybe the attendees can ask them on the chat box. If you can see it, because obviously some people are using the chat, so that may be a way around it, but I'm not sure why you cannot see the Q&A box. Chat is working and Q&A is not working, apparently, according to Mike Shattuck.
Oh, there's a Q&A one.
Oh, there we go.
Oh.
Now getting the Q&A box as well.
Okay. There's a very good question. Could the biological function of PCSK9 change depending on whether it is found on HDL versus LDL? And again, I think this is an excellent question. Now we're looking at this in postprandial samples. And indeed, it seems to be the case that there may be a shuttling of PCSK9 between the different lipoproteins. And I think this is very exciting, because there is evidence that PCSK9 binding to LDL inhibits PCSK9 activity. And now the question is, what's the role on HDL? How does HDL affect PCSK9? And I think it would make a lot of sense if the distribution and the concentrations of the lipoprotein particles impact on PCSK9, which is actually known to regulate the circulating concentrations. So indeed, I think this is a is a very plausible idea that the compartmentalization is not static, but the compartmentalization may change whether you're in the fasting state or in the postprandial state.
I think there's also a question from Lucy Carrier. I can see. I don't know if you can see that.
Yeah. Hi, Lucy. So Lucy asked, high levels of HDL are also found in alcoholic patients. Is there any studies with anti-PCSK9 inhibitors? So I'm not aware that a study was done in alcoholic patients. I think Thomas Eschenhagen probably would know. But I think, of course, the raised HDL levels in patients with alcohol consumption in part is also a response to the liver damage. And again, it shows that, not all elevations of HDL are necessarily always protective. Now, David Eisner said, I can get the Q&A in there. So I'm switching, maybe I can, good, okay, I've seen it here. Yeah, so I think there's many questions to be asked. I mean, the PCSK9 inhibitors clearly show great promise, and I think we look particularly forward to these antisense therapies, because right now the PCSK9 inhibitors, you give a monoclonal antibody injection every couple of months, but the promise of antisense technology, of course, would be that maybe you don't need the frequent injections and you have maybe a more sustained response in lowering your LDL cholesterol.
So there is another question on the chat that I can see, just the last question from ThinkPad. Don't know if you could answer this one as well, Manuel, if you can see it.
Let me see.
So somebody would like to know.
Yeah. Okay, so I think, again, of course, many omics approaches are used. I think what has been done quite extensively now, and for example, is also done in UK Biobank, is this NMR-based approach, because this NMR-based approach for lipoprotein profiling gives you around 200 metabolite ratios. It's relatively cheap and it can be done in large throughput. One of the bottlenecks for the other analysis is that it's not that automated. It's still relatively expensive. But we see now more and more studies that actually use different technologies, not just mass spectrometry, but like multiplexing, antibody assays, or aptamer assays to interrogate the plasma proteome. The only thing I would like to say here, and I think this is a word of caution, is that again, we are relying on binders. And the more binders we are multiplexing, the more questions are asked whether they are specific, whether they are actually confounded by certain variables that we don't appreciate, and what is the dynamic range of these proteins in the circulation. So are we actually measuring in the right dynamic range? And in particular to aptamer technology, which are using oligonucleotides, you can measure many proteins based on these binders, but there were concerns that were raised about the specificity. So I think it's a good discovery approach, but like any discovery approach, you should perform validation. And you should not just rely on the binder, but you should actually provide independent measurements to confirm that this finding is true. But clearly what we are going to see is we are having more and more protein measurements that will help us in the cardiometabolic phenotyping. So we will move on from just measuring ApoA1, ApoB, and maybe CRP to now measuring hundreds of proteins, either by mass spectrometry or by aptamers or by other antibody-based technologies. So we get a new molecular phenotyping, and I think this will provide really novel insights into how certain cardiovascular diseases manifest themselves at the plasma proteome level.
Yeah.
Are there any post-translational modifications to PCSK9? There's another question, I just saw this one. Are there any post-translational modifications to PCSK9 that can be responsible for preferred binding to LDL versus HDL? Again, I think this is an excellent question. What has been shown, and this is really nice work from the group in Canada of Thomas Lagasse, and I think they've just published another paper in JBC where they have looked at mutations in PCSK9, so not that post-translational modifications, but gain of function and loss of function mutations that have been shown for PCSK9. And what they could nicely show is that this gain and loss of function mutations of PCSK9 alter the binding of PCSK9 to LDL. So it is very plausible that we could also have an effect of these mutations on PCSK9 on HDL binding, and that depending on what variant you have, you may have preferential binding of PCSK9 to LDL versus HDL. But again, I think this is, of course, all speculative, but for us was really striking that HDL indeed seems to be the main carrier of PCSK9 in the circulation. And this, to my knowledge, has not been studied to date.
There's also another one that's just come from anonymous attendee on the Q&A. I don't know if you can see it as the last question, but to some extent you probably answered it before, but...
in the Q&A one?
Yes.
Okay, this is again a question of, does PCSK9 binding to HDL modulate HDL expression? That might explain the cardioprotective factor you were talking about. Again, excellent question. I wish I would have access to the lab right now, which we don't. But yeah, clearly, I think, yeah, this is the next step. We would like to see how PCSK9 binding to HDL not just affects PCSK9 activity, but also how it crosstalks with ApoE1, for example, expression or HDL expression in the liver. So these studies all need to be done. Like I said, this is pretty much hot off the press. It just appeared in JACC last week. And many of the studies that have been done for PCSK9 and LDL, I think we now need to look at in the context of HDL. And as was pointed out earlier, not just in fasted individuals, but also in postprandial people, to better understand how our physiological response actually changes PCSK9 activity. Because so far PCSK9, I mean, is an excellent treatment target, but it's still not quite clear what's the physiological role of PCSK9. Because right now the main benefit is if we inhibit it, because we get very low LDL cholesterol levels. So clearly there must be a reason why PCSK9 is secreted, and there must be also a mechanism how the lipoproteins regulate the activity of PCSK9. As another question, will fasting status influence the results of proteomics? Yeah. So usually we only use fasted plasma for proteomics. And again, the reason is that you want to have some consistency. And when you have non-fasted plasma samples, you never quite know when the last food intake was. And of course, you have this major confounding by all these lipids in the circulation. Now, many of you who work with peptides, you know that peptides, of course, we isolate them based on their hydrophobicity. So if we have a lot of lipid contaminants in postprandial samples, often that can interfere with some peptide measurements. So in fact, we did some proteomics on postprandial samples, but the background in plasma is not equal because during the postprandial phase, the lipid content varies. And that to some extent, if you are not very careful and if you don't use extra purification steps, then the lipids in the postprandial samples may interfere with some of your peptide measurements. So this is something you just technically you need to be aware of. When we perform the same assay in a different matrix, we need to be sure that the matrix doesn't impact on the assay. And if we use postprandial samples, the matrix changes every hour because first the triglycerides and the lipoproteins that contain triglycerides rise, and within a couple of hours they are removed. So we really must be sure that if you perform actually any measurement, not just mass spectrometry measurement in postprandial samples, that your measurement is not confounded by the varying lipoprotein content in your sample.
Great. I mean, I think, shall we, I mean, there is one last question in the chat. I don't know if you can actually respond to this. It's a very specific question. And I think probably we should stop then after that one.
Okay. Now this is, okay, no, I don't know the answer to this question. Is the relationship of ApoB 100 and PLIN2? I'm sorry, I have to skip this one, but I'll look it up. There's another one which is easier. Can you use plasma samples frozen at -80 for mass spec? And yes, absolutely you can. You should use samples that are stored at -80. The proteome, unlike RNA, is very stable, so you can actually use samples that have been stored for several years, but they should have been stored at -80 rather than at -20.
Right. Now, Manuel, I mean, I think we'll stop here. You kept the time perfectly. Thank you very much for this fantastic webinar. Manuel has kindly agreed for this to be recorded as well. So as soon as possible, this will be uploaded and there will be an Zoom follow-up email with the link where you can view the webinar. But we have created a ISHR cardiovascular webinar channel as well, YouTube channel. So all the videos will be available there. I mean, this only applies, as I said, to people that have agreed to have their webinars recorded. So thanks very much. I think we close a successful first week of the ISHR Cardiovascular webinar series. Next week, we start off with Tish Murphy, and I will be sending out invites for those, but please use the spreadsheet, the Google Doc spreadsheet, to register. And also there are still, I think, 3 places left until the end of June. So if anybody does want to give a talk, you've got 3 spots left. So thanks again for everyone. And I would like to, first of all, lastly, thank the ISHR and just share this slide with you, if you don't mind. And you will get some information there on how you can join the ISHR should you wish to. And you can also get that information on the spreadsheet, Google Doc spreadsheet. So thank you once again to everyone. And I will probably stop sharing now. And Manuel, thanks again, and have a lovely evening.